In the world of medicine, some of the most powerful diagnostic clues are actually hidden in plain sight, circulating right in our own blood. Today, we're going to dive deep and unlock the secrets of clinical enzymology. We'll explore how these tiny biological engines, which honestly power every single aspect of our lives, become these critical messengers when something goes wrong in the body.
So we're going to tackle this like a detective building a case, step by step. We'll kick things off with the absolute fundamentals, getting to know our key players, the enzymes. Then we'll learn the language, how we classify them, and how we measure them. From there, we'll see how these enzymes work inside the body before we put it all together to solve some real clinical puzzles in our case studies.
So let's start at the very beginning. What exactly is an enzyme? Let's strip it back to the basics in Section 1 and really get a handle on the critical role these molecules play in the chemistry of life itself. At their core, enzymes are basically nature's ultimate facilitators. These proteins are biological catalysts. So what happens is, an enzyme binds to its specific target, which we call the substrate. This forms an enzyme-substrate complex that stabilizes the reaction's transition state. Think of it like this. Every chemical reaction has an energy hill it needs to climb to get started. Well, enzymes come along and just drastically lower the height of that hill. This lets reactions happen millions of times faster than they normally would. And the really cool part? The enzyme itself comes out completely unchanged, ready to go again.
Okay, so understanding what enzymes do is the first step. But to actually use them in medicine, we need a common language and a standardized way to measure them. And that brings us right to Section 2, where we'll get into the systems of classification and measurement.
So with just a countless number of enzymes out there, how do scientists keep them all straight? Well, the Enzyme Commission, or EC, came up with a really brilliant solution, a unique 4-digit code for every single enzyme. Let's break down the one for creatine kinase, EC 2.7.3.2. The first number, the 2, tells us it's a transferase. It moves a functional group from one molecule to another. The 7 gets more specific, telling us it's a phosphotransferase, so it's moving a phosphate group. The 3 tells us the thing accepting that group is a nitrogenous group. And finally, the 2 is its own unique identifier. It's kind of like a biological Dewey Decimal System.
So how do we quantify enzymes in a blood sample? I mean, we don't count them one by one. Instead, we measure what they do. We measure their activity. The gold standard for this is the International Unit, or just U. 1 U is the amount of enzyme that's needed to convert 1 micromole of substrate every minute. So when you see a lab result that says AST is 300 U per liter, that's not a direct count of the enzymes. It's a measure of its catalytic power in a liter of serum. And from that, we can infer the enzyme's concentration.
All right, now let's explore the diversity of enzymes inside our own bodies. Turns out, a single enzyme can actually have multiple different forms, and understanding these variations is absolutely key to pinpointing where a problem is coming from.
Okay, 2 really important terms you have to know are isoenzymes and isoforms. They sound similar, but they're not the same. Isoenzymes are different versions of an enzyme that do the same job, but, and this is the key part, they are coded by different genes. So for instance, the alkaline phosphatase that comes from your liver is genetically different from the one in your bones. Isoforms, on the other hand, all come from a single gene. They are just variations of the same protein that get created by modifications made after the protein is built. The subunits of creatine kinase are a perfect example of this.
This diagram here really shows how isoenzymes get created. Just imagine you have 2 different genes, A and B. Each one produces a different protein subunit. Let's call them A and B. Well, these subunits can then mix and match to form different combinations. You could have 2 A's together, 2 B's, or an A and a B. This whole combinatorial power is what gives us multiple distinct isoenzymes, and each one has unique properties that turn out to be incredibly useful for diagnosis. With all that theory in place, it's time to bridge the gap and head into the clinic. In Section 4, we'll see how we use these enzymes in real-life diagnosis, and we'll also confront the single biggest challenge in all of clinical enzymology. This slide right here lists the all-stars of clinical enzymology. These are the main enzymes you're going to see measured in blood tests all the time. You've got ALT, which is a key indicator of liver cell damage. ALP points towards either bile duct problems or bone disease. AST can mean either liver or muscle damage. CK is for muscle injury. GGT is a very sensitive liver marker. Amylase and lipase are the go-tos for pancreatitis. And LDH is a more general marker for cell breakdown, which you see in things like hemolysis or even cancer. Now, this slide is maybe one of the most important concepts to grasp because it shows us both why enzymes are so useful and what our biggest challenges.
Look at these concentration gradients for AST, ALT, and CK. And really pay attention to that y-axis. It's a logarithmic scale. This means these enzymes are thousands, sometimes tens of thousands of times more concentrated inside our cells than they are floating around in our blood. This is why even a tiny amount of cell damage can cause a massive relative spike in the blood level. But it's also our biggest problem, a lack of tissue specificity. If AST is high, where's it coming from? The heart, the liver, skeletal muscle? That's the central puzzle we're so often trying to solve.
All right, let's put all these principles into practice. We're going to put on our detective hats and analyze some real-world case studies involving liver enzyme abnormalities and see exactly how we solve that puzzle.
So when we look at liver enzymes, they generally fall into one of two main patterns, or buckets. The first one is the hepatocellular pattern. This is when enzymes like AST and ALT are way up. It tells us that the main problem is damage to the actual liver cells, the hepatitis. The second pattern is the cholestatic pattern, and that's dominated by high ALP and GGT. This points to a problem with bile flow, some kind of blockage or obstruction. And of course, sometimes you see a mix of both.
Okay, let's look at our first case. Picture this, a 56-year-old man who has a known history of heavy drinking is found semi-comatose. When he's examined, he's jaundiced, his abdomen is swollen with fluid, we call that ascites, and his liver is enlarged. The whole clinical picture is just screaming decompensated alcoholic liver disease. Now, let's see if the labs agree.
And here are his results. We see a classic mixed pattern. His AST and ALT are way up, 317 and 295. That's the hepatocellular damage right there. But his ALP is also up at 175, which shows us there's a cholestatic component too. And then look at his albumin. It's really low at 24. Since albumin is a protein made by the liver, this low level tells us the liver's ability to build things. Its synthetic function is failing. The labs just perfectly confirm the clinical diagnosis.
Our next case is a 59-year-old woman. She has stage 4 esophageal cancer that has unfortunately spread to her liver. An imaging scan showed that a large cancerous lymph node was physically blocking her bile ducts, and this led to a diagnosis of cholestatic jaundice caused by this malignant obstruction. Now, look at these lab results. They paint a perfect textbook cholestatic picture. Her alkaline phosphatase is a staggering 640, and her GGT is also extremely high at 630. But meanwhile, her ALT, which is the marker for liver cell damage, is only up a little bit. This huge, disproportionate rise in the cholestatic enzymes is the absolute signature of a biliary obstruction, and it matches her imaging findings perfectly.
Okay, here's a real puzzle for you. Case 4 is a 40-year-old woman who was found to have a persistently high ALP on some routine blood tests. But everything else was totally normal. All her other liver enzymes were fine. They did an extensive workup checking for viruses, autoimmune diseases, imaging, and everything came back negative. So the million-dollar question is, if it's not the liver, where is this isolated high ALP coming from?
Well, the solution lies in isoenzyme analysis. By separating out the different forms of ALP, we get our answer. You can see her total ALP is high at 173, but her liver and bone parts are actually normal. The real culprit is the intestinal ALP, which is way up. This is a known, totally benign phenomenon that's common in people with blood groups O and B, especially after a meal. It's not clinically significant at all, and just like that, the mystery is solved.
In our final section, let's broaden our scope a bit beyond the liver. These next few are quick-fire cases that are going to show you just how versatile enzyme analysis is for diagnosing a whole range of conditions all over the body.
So here are 3 quick examples. In case 5, a woman had a high ALP of 416. The cause? Severe vitamin D deficiency, which revs up bone cell activity. As soon as her vitamin D was replaced, her ALP went back to normal. Case 6, a 19-year-old guy had a creatine kinase over 1,000. It wasn't a medication side effect, it was just from his super strenuous daily gym workouts. And finally, in case 7, a patient with lymphoma had an LDH that was literally off the charts, over 3,000. This was caused by the massive, rapid breakdown of tumor cells.
Let's finish up with one last diagnostic puzzle. Step 1. A 22-year-old woman gets a routine blood test, and it shows a really high serum amylase of 571. So your first thought is pancreatitis. But step 2, clinically, she has 0 symptoms of pancreatitis. So a follow-up test is done, a urine amylase. Now, amylase is the only enzyme small enough to be filtered by the kidneys, so we'd expect it to be high in the urine too, but hers comes back completely normal. Step 3, this discrepancy, high in the blood, normal in the urine, that is the key clue. It points directly to the answer, and in step 4, the diagnosis of macroamylasemia is made. So what on earth is macroamylasemia? Well, it's a fascinating and totally benign clinical pitfall. In this condition, the amylase enzyme actually binds to a big antibody in the blood. This creates a complex that's just too large to be filtered out by the kidneys. This means the amylase can't get cleared into the urine, so it just builds up in the blood, giving a falsely high reading. It's a harmless condition, but it's such a powerful reminder of why understanding the biochemistry behind the numbers is so incredibly important. It shows how a deep dive into the science can turn a really worrying result into a resolved puzzle, preventing a lot of unnecessary tests and anxiety.